Hi, everybody. I'm very happy to tell you about my research on the geometry and role of sequential activity in olfactory processing. So as a bit of a departure from yesterday, I'm a physiologist in a lab that studies smell or olfaction. And so to better understand the neural underpinnings of olfaction, we focus on mice. So mice rely on olfaction to navigate the world based on faint transient whiff of a few molecules, which you can't see, but believe me, they're there. Mice need to detect and correctly identify the presence of objects in the environment. Ooh, that's horrendous brightness. Sorry, I think that might be the effect of turning the brightness all the way up, is that the colors are gone, but whatever. Yeah, but often they have to do this at a distance and plan appropriate actions, like navigating to what they want. So how do mice solve this? Well, mouse is gone. Yeah, sorry. Do you mind if we just do the brightness? Because there's a lot of color stuff going on here. Okay, great. Sorry. So how does the olfactory system perform this feat? So just really brief overview of the anatomy. Odor molecules come in the nose when an animal inhales. They bind to receptors on receptor neurons in the olfactory epithelium in the nose. Those neurons project their axons to a place in the brain called the olfactory bulb. The structure is called glomeruli. The olfactory bulb is kind of like the olfactory retina, where there's a lot of processing that happens internally. The output is then sent from the mitral and tufted cells to the cortex. And so we, in the lab and in my work, we focus on the properties of these cells because that represents the output of the transformation of the olfactory bulb. So unlike the retina, we don't know the features that the olfactory bulb is built to find or represent. And so there's been a couple of schools of thought in looking for maybe how the activity is organized in the olfactory bulb and what receptive fields would look like. So a classic way to study this is using something like two-photon imaging, where you can present odors to a mouse while you look at its brain and see that different sets of neurons, these are just same neurons, but different ones activate for different odors. And that effectively tells you which neurons activate for these odors, but due to the kind of slowness of imaging, you kind of lose the temporal component. The other side of the field, and our lab uses electrophysiology, and here we found really interesting feature of the way these cells respond to odors, which is that despite the fact that an odor might last for seconds or more, the responses of the neurons are tightly timed to a particular phase of the animal's breathing cycle in tens of milliseconds bursts. And that's repeatable over and over again every time they smell the odor. And so using this method, we can see at what time these neurons respond. If you look at lots of neurons and lots of odors, you can see that you'll see odor-specific sequences that tile the respiration cycle when an animal breathes an odor. And so that got us very interested in maybe trying to find out what defines this order, the order in which mitral cells will respond to an odor, and what information is encoded in these sequences. So we want to understand how the temporal coding part of olfaction relates maybe to the classic weight coding and
So John, just a question there.
Yes.
So I'm not seeing the slides sync to the expiration.
So that's it's upside down. So in this case, it's breathe out, breathe in, and then they hold their breath, breathe out, breathe in. They breathe opposite to us when they're awake. So they go, and when they're anesthetized, it flips or asleep. Weird stuff. Okay, so we want to marry these abilities to look at which neurons are active over time, as also how to see the high resolution response timing that we get from EFIS. So luckily, there's GCamp 8F to the rescue. There's a new calcium indicator that gives us basically electrophysiology resolution in terms of time. So now within the same group of neurons, we can see how they respond to lots of different odors, like who responds, and also exactly when they respond within a sniff. And that's what I'm showing you there in the bars. If you change odors, different neurons respond at different times. So you see the reorganization when you go from epitheliate to two-epitheliate. These are generic odors. Okay, so what you see is that you have a reorganization of which neurons respond when you give a new odor. But if you look at if you order them by the latency of activation, you can see that you have another sequence. So basically, each odor produces a sequence that's just across different cells. So we want to use this ability now to try to understand maybe the shape of tuning across these neurons for odors. So one way is to take the way we approach this is to take the response of one neuron across many odorants and measure some feature of the response, such as the an average of the activity during the first sniff. We can arrange these numbers in a matrix where we have the response of each mitral cell across every odor. So we get this picture, which is really rich, where now you can try to see whether there's trends here or relationships between the way the mitral cells are tuned. And to approach this, we look at the pairwise correlation between the mitral cell tuning vectors, so the columns of this matrix. So if you look at the Pearson's correlation, you can see a picture that emerges, which should immediately jump out to you, is that you have a somewhat modular structure, so groups of neurons that are highly co-tuned, but never exactly the same, as well as other groups of neurons that have maybe opposite tuning. So it seems like there's maybe some shape here, which, in order to visualize, we resort to our old friend dimensionality reduction. So in this case, we're using multidimensional scaling, which is basically PCA for distance matrix. What we see in the gray dots there is that you can take all of the mitral cells and embed them into a lower dimensional space where it tries to preserve the differences in their tuning as best it can. And what you can hopefully observe is that you get some kind of smooth tiling of what appears to be kind of like an odor tuning manifold here. So but does this reproduce any interesting structure that we saw on the right? Well, one way to think about it is we can color these dots however we want. One way would be for these cells, which odor activates them the most, and see whether there's some organization there. And what we see is that you can reproduce to some extent the modular structure on the right. We'll see groups of cells that will share some max tuning, but that they continuously vary along this hole.
So, sorry, are you in one glomerulus at the moment?
This is across like a big chunk of mitral cells. So this is across many glomeruli.
Yeah. Okay. Fine. Cool.
Yeah. So this is where you
sort of the glomerulus.
You can. We do it now. Here we don't have the labeling because it's different layers. Now in the lab we can actually stimulate all the glomeruli and find who's connected to who. But this was a little earlier. Yeah. Basically, some of these little guys are almost certainly coming from the same glomeruli.
Yeah.
Like those they're sisters, we call them. The big ones are maybe not. Okay, but basically we can see this kind of smooth variation here. So okay, so we can here I can do it again for another mouse here, believing that we can reproduce it with other sets of cells. So here basically I showed you maybe there's some shape in the way that the tuning is kind of arranged. But I've ignored so far the temporal component. We're just looking at kind of set membership and rate. But we can look at the timing of when cells activate relative to when the animal inhales using the method and ID the onsets. So if these are cell traces, you can see when each cell crosses a threshold and say that's when the latency of the cell's response was. Using that information, we can actually color those dots instead of with the max tuning. We actually color them with the latency of response for a given odor. So we can watch how odor responses propagate through the space. So now we can actually see the temporal component superimposed on this this kind of rate-based structure that from the event. And so what you see is that for example, this odor 3-heptanoate, the odor seems to activate the cells on the right and propagate to the left. If you with the later ones on the left, so the yellow being the later part of the sniff. If you change the odor, these waves seem to propagate from different angles. And so you see this as you change the odors, you basically hit your manifold from different angles and travel through it in some kind of smooth way. If you have more cells and you project in 3D instead of 2D, you can see that your odors will cut through your ball of tuning embedding space. Here you can see it again maybe a little bit more obviously with ethyl tiglate here. So you're starting and radiating out. So it's kind of giving you this view that your odor responses involve activating some initial set of neurons and then it travels as a function of dissimilarity in tuning. Yeah, so the trick is that all of the mitral cells, as far as we know, are only coupled with inhibitory connections. So there could be things like disinhibition, all sorts of ways they can interact. We don't really, that's still a bit undiscovered in the field, but we're working to try to see maybe if ones across the manifold, for example, inhibit each other, or that maybe supports the space. So we have ways to quantify this, not to belabor it, but basically we can take our projection here. You see the first neurons that activate and later ones in the middle and the end of the sequence. And we can relate the difference in latency for each odor response to the distance in tuning in the space. And what we find is a linear relationship when we average over odors. And this is for one mouse. If you look across many mice, they all seem to obey this rule. And if you look at the group average, we can basically estimate the time constant of the wave propagation through the tuning space. So we can see that this is kind of a steady march through the tuning space, not dependent on odor. It seems to move the same way with the same speed. So what we can tell from this is that sequences are organized, but we want to know what do they mean, right? So that's cool that maybe it's organized, but what does this buy you computationally? And so to respect our time limits, I insist that you come to the poster and find out. As a brief preview, what you can expect is that we can cut up the sequences and say, what happens with the early activity? Maybe there's some role there. And what we found is that the earliest parts of these sequences seem to be concentration invariant. So the first cells to respond seem to respond as you change the amount of the odor that the mouse is smelling. It just grows in how far the wave propagates, but it starts from the same angle. And if you look at the later part of the sequence, we have constructed a model that posits that this later part can be used to train downstream readouts, for example, the piriform cortex, the main output target, using a very simple Heppian tool that can learn the appropriate weights to permit generalization. So how you can smell something you've never smelled before and know that it smells similar to other things that you do know without having to learn how to smell everything for the first time. So with that, I'm going to do.